in essence, in mechanical transduction, what you're envisaging, and sorry for the slightly naff guys in suits having a tug of war, but you've got this interplay between the extracellular forces, the movement and things changing outside the cell, and the cell's force generation machinery. And depending on these balance out, these pull and relative like tug of war, and these switches, these 13 tailing switches, sit in the middle of that, and they respond to changes from inside and outside the cell. And this enables constant force feedback and enables the behavior. What I came to, this was back in the day where you're allowed to travel and go to conferences. When I sat at an airport in Sydney, I started to think about this as a very simple question. But what happens if this extracellular environment is constant and predictable? And I'm showing that as a wall. What I'm not saying here is that this is stable and never changes, because I don't think that's the case. But in a lot of our cell types and in a lot of our tissues, the environment is incredibly predictable. It's constantly maintained, it's constantly repaired in such a way by the cell itself so that the cell knows what that is. So if this is a predictable thing, then here we've got a very different scenario. We're not, the switch pattern of the tailings isn't dependent on two varying things. What we've got here is that the extracellular forces can be largely known. I wouldn't say constant, but known. It's no longer a tug of war. Here what we've got is we've got a mechanical device attached at one end, which the cell can pull on and generate forces to cause these transitions and flick these switches internally. So here we've got a switch box which the cell itself can control to coordinate its function. And here, so yeah, the cell now generates forces and alters the switch patterns. So it was that finding, which that theoretical shift in my thinking, which led to this paper, which has been under review since, I don't know, I was a young man when it first went in, it seems. But this is a theory I've been developing, trying to understand what would happen if this was predictable and if this could occur and be stable. And that's what I want to talk about for the last 10 minutes or so, is this theory and this idea to get feedback on this. And it's this, I mean, if the cell can coordinate this, then the cell, a signaling pathway, could trigger contractility, which would flick one switch. Because these switches are individual and they're quantized, then you could flick these individually and control their pattern via intercellular. So now how I start to think of tailing, and a lot of work we're doing at the single molecule and the theoretical level, is to consider that tailing and these mechanosensitive events I've been talking about, this is actually information which has been stored in these, because we talk about these domains going from a folded to an unfolded state. Both of these are thermodynamically stable, so you can switch between that to that with mechanical force, or reduce the force to go back to that. So if you think about this is actually a binary switch, then this is a folded state, which is O, but you can pull on that tailing and trigger it and convert one of these domains to a 1. And here's a small example. If you had 3 domains, you exert force on it, you transition 1, and then the other 2 would stay folded. And then you could either reduce the force and go back to that, or you could pull further and unfold another one. But because each of these have got their own unique mechanical response, the order which these will unfold when you pull on it is quite reproducible. And because of the feedback, because of what's recruited, then this happens with incredible robustness. So what you could start to imagine is that what we're actually really looking at is a binary string, which in the cytosol is all in this folded O state, which is what we did all of our structural work on. But once it couples to the integrins at the and makes these adhesion complexes and connects to the actin cytoskeleton, then they can adopt a certain pattern of these switches, which coordinates the cell how to behave. But from that position, you can then generate contractility. Say a signal generates an increase in rack activity, or you generate contractility. You can convert one of these switches, changing this string of information, and you might pull again and change another. And then you might have a decrease in contraction, or you might have proteins diffusing away, which lead to a reduction, and then the pattern can change again. But all the while, this is information that is being stored in these molecules, and this is information which is coordinating the cell how to function. And I think this is what's happening in those differentiating stem cells. But I think it's information which is occurring. And the reason I think that, and what we can show, is we can show that tailing is an information storing molecule, because we can do single molecule studies on it, and because we now know the unfolding and refolding kinetics, we can simulate this in silico. But what this enables you to do is to hold a tailing molecule, and if you hold it under tension, then you can add numbers to it by pulling on it, or reduce numbers by letting a contraction. And as long as you hold both ends of this molecule, it will remember that information. It will remember the shape it's in indefinitely. And that means you can do sums on it because it will remember what state it was already in, and then you can add more to it by pulling on it and doing these switches. A big assumption here is that this only works if the mechanical environment is perfect and you've got a perfect tether. If it's constantly moving, then that it's going to still store information, but it's going to be very noisy and not very persistent. But if it is predictable, and this leads to a prediction which arises from this theoretical thinking, is that the mechanical environment of the brain is perfect. And for a long time, mechanobiology in the brain and mechanotransduction, because the brain is soft, it's been considered not to be particularly effective for mechanotransduction. But this is actually a strength, because this means because it's soft and because it's not that stiff, then that means that there's the opportunity, and the theory predicts that every single synapse in the brain will form its own extracellular matrix around the edge to form a perfect mechanical unit. And this is borne out because if you look at any sort of neuronal process, the ECM is constantly undergoing maintenance and remodeling all the time. And I think that's to make this a perfect mechanical environment. Because if the mechanical environment is perfect, the prediction is, and what I'm arguing here, is that mechanical computation can be perfect, and that you can generate these same responses every single time and with high fidelity. And the reason to go back to the strapline in the title is that if you look at what a synapse looks like, and apologies for my poor artwork, this looks like more like a nostril than a nose than a synapse. But if you look at how these things are organized, then you've got the active zone and all of the classical neuroscience, but you've got round the edge, you've got the ECM components, and you've got the integrins, and you've got the actin cytoskeleton. So the integrins, you don't normally get an integrin which hasn't got a tailin attached to it, because tailin regulates the integrin activity. But the tailin's been neglected just as a linker. But if you look at the literature on integrins' role and ECM's role in pretty much all neuronal processes, it's quite well established. But what we've got, you can start to re-envisage this as a mechanical unit. Around the periphery, what you've got is you've got an array of switches in these scaffolds, showing cartoon here. And this, you've got to remember, this would be decorated with hundreds and hundreds of signaling molecules to give rise to the adhesion itself. And to show this slightly more conceptually, if you imagine that all of these switches, I've drawn them in this different array here with a tailing molecule with 13 switches running down the page, then in an adhesion, you might have 100 tailings. Then there'll be all of these different patterns of ones and O's, depending on whether each of these domains is folded or unfolded. And the hypothesis I'm generating here is that electrochemical transmission across this site causes a contraction. It's not, these things aren't under high force, they're under low force. That's how you store this information. But when you signal, you get contractility, a short burst of it, and that changes the pattern of these switches in that synapse on both the pre and the postsynaptic side, and potentially in any supporting roles as well. So as you signal, you could be constantly updating these switches to change and tune how each of these synapses behaves. And again, one of the things we're trying to do with Melissa is to establish how many of these molecules are actually in these synapses. I mean, it's not a given. Integrins are definitely essential and they're there, but how many? If you assume 100 tailings per synapse, and 8 of these switches are able to be used in this, then you might say there's 100 bytes of information could be written into these scaffolds. But what this means is the hypothesis I'm generating is that memory is mechanical in nature, that this binary coding of these mechanical switches, which hold the synapse together, is actually where information is stored, and the electrical and chemical signaling and transmission across this updates this coding in both the pre and the post-synaptic sides, altering the information in that synapse, but also altering the activity of that synapse. And something which is incredibly speculative, even more than that, is that every single cell in our body, apart from the ones which aren't attached, but 99%, nearly all of the cells in our body, absolutely require integrin adhesions. So every single cell has got this mechanical coding in it. So you could start to envisage that this is a mechanical code, a mechanical machine code, which coordinates this whole organism, any organism. But one of the things which is really important is that this is all well and good. You can get on board, and as one of the reviewers very vehemently said, he could totally understand that this is a memory molecule and that you could store information in a single molecule, but that doesn't explain how you could read this. How could the code be read? So I just want to have a couple of minutes just to highlight a few ideas of this. And this is the reimagination of the tailing code we discussed here. Apart from talking about it in terms of a binary string, folded, 4 folders, 000. So the readout is the actual proteins recruited to that site. And the hypothesis states that the number of tailing molecules in a neuron is probably quite fixed, and they're distributed across the whole of the cell in all of the synapses. So there's a finite number of each of these switches. So the relative amount of the switch in each state, in each synapse, will determine where these soluble diffusible molecules reside. So as you change the shape of this, each time you generate contractility, you change the information in that molecule, and which is read out by the proteins which are now decorated on that molecule. And something really important to point out is if you pull on a molecule where one of the domains is already unfolded, you can't re-unfold that same domain. So if you pull on that tether, then the only thing you can do is to unfold another domain. So you can have this change in these patterns depending on what state that molecule is already in at the start, if that makes sense. So in terms of cell migration, then this contractility dependent growth of adhesions is really well established. You apply contraction onto an adhesion, contractility on an adhesion, it grows, it recruits more molecules. You don't stimulate it and it atrophies and shrinks. And this would be the mechanism for how this would be working in the synapses. If you're stimulating a synapse, the adhesion would grow and the signaling molecules recruited would be different, and you'd get a stronger signaling. And you would redistribute the ligands across the whole of the synapse, which I'll show on the next slide. So what this would mean is that a stimulated adhesion would have a different pattern of folded and unfolded domains to a unstimulated. So the relative distribution of all of the signaling molecules would be different depending on the pattern and whether that synapse has been stimulated. So what I'm trying to do from here is just to highlight there's lots of things known from what I can gather. So I'm new to the neuroscience field, but there's lots of things like synaptic tagging and synaptic capture and probability of vesicular release. And all of these are known to be sophisticated and involved in these processes, but the precise mechanism is quite hard to pin down on what's going on. But one possibility, and this is quite speculative, but just to suggest the utility of this as a framework, is that the hypothesis would be that a stimulated synapse, you could have a contraction, again, sticking with this same format, these arrows. This this shows a cytoskeletal contraction. You could have a neuron where it triggers one of these, it pulls on this synapse, causing a conformational change, which causes this synapse to fire compared to this synapse where the contraction wasn't there. And that would cause a conformational change in one of these switches and change the signaling and change the release. So this could be a mechanical basis, because it's very clear from the recent papers on this that actin contractility is essential for vesicular release. It might be that it's via this change in this mechanical coding. But something which is better understood as a concept but not understood as a mechanism is that when you do stimulate a synapse, you start to get this thing called synaptic tagging, where that synapse is marked in some way in that it recruits specific molecules to that site compared to other sites. And in that scenario, you get, it could easily be explained from this. If you have a switch in that synapse, which is unfolded, you create a new binding site for a ligand, then only the stimulated synapses would have that tag, that exposed binding site. So you'd recruit more of this blue protein to that site and relative to the unstimulated. So these synapses would be talking to each other. This pool of proteins would be distributed differently across the whole neuron, depending on which synapses were most active. And another thing is inverse synaptic tagging, where you have things which seem to dampen the potentiation of the synapse. And the same thing could happen here. If they're on the unfolded state, then they could be released by stimulation and then go and sit and occupy unstimulated ones. So you can have this constant crosstalk between these different synapses to give rise to this. Okay, I'll try and wrap this up. And then building this one step further, as these switches, the history of them is locked in. Like if you've already got one pattern, then a pull on it will change the pattern again, built on that previous pattern. Then the pattern of switches in the synapse and in the neuron and in the whole circuits would be constantly updated based on what the patterns already were and what those adhesive complexes and what that synapse already looked like, which again, I think is pretty well established. But here it's a physical biochemical basis for how that might work. And yeah, I need to get a move on. So what this leads to is it leads to a model where if all of these synapses are constantly changing these patterns of switches, which coordinates their activity and the information, that if you have a circuit, this is just a textbook example of a reverberating circuit, but I've put on these MeshCODE patterns, then you could imagine a scenario where you stimulate a circuit and it causes the synapse to fire, which updates the patterns in both the pre and the post-synaptic side, which causes all of the neurons to fire. Like this is well established that this happens, but quite how and which ones fire, I would say, is less well established. But here you'd be constantly changing these patterns, and it would only be when the whole thing balances back out, all the mechanical forces sort of cancel out and become balanced again, would this stop. But once it did stop, then you would have a circuit where all of the synapses would be updated with this new information. And it might be that a future input would immediately trigger the correct output if it's learning, or it might constantly be updating this information in these circuits. So this gives a framework where you could imagine that these binary, all of this inputs into an organism are mediated through a sensory, some sort of sensation, which triggers an action potential spike train, which sends that information inwards through these circuitry, through these nerves, to the brain. In this model, you could begin to imagine that every single synapse and every single connection here would be constantly being updated with new information as this changed. It would change each individual synapse in a highly dynamic fashion. And then that would mean that the organism would be constantly learning and updating for the best probabilities based on the future signals. And the reason this could work is that because this is a binary code, these spike trains of a binary signal would just be updating this register. So I'm not saying I understand this, but just as a framework or as a theoretical concept of this. How long have I got left? Have I got to stop? If not, I've got about.
If this is your conclusion slide, then we're set. Okay.
Yeah. Well, no, it's not actually. Let's skip through that. Okay, so I'll skip through all of that. I just want to wrap it up because there's a lot of possible framework theoretical conclusions from this. You could start to imagine an explanation, a mechanical explanation for many different things. And just to ground this back on our main research in the lab, is that all of this comes because we've got these molecules which have got molecular memory, and the shape of them will determine how they behave, how the cell behaves, how the tissue behaves, and here how the organism behaves. And that it can store this information persistently as a binary string. And because it's based on really well-established biophysical principles, I think that this has identified a physical location for data storage in humans and in all animals. And as I say, I think this also hints that this is a machine code. All of the cells are running the same mechanical code, which talks to the genomic code to make them all work together as a unit rather than as a load of individual cells. And my argument is that this mechanical layer. So, and the other important thing to say here is that so far this is completely. It complements all existing frameworks for neurofunction. And I would say that the mechanical layer would integrate with the well-established chemical and ionic signaling layers to provide an underlying mechanism for information processing and storage. And I would also argue that by converting sensory and temporal inputs into a binary format, this might identify an addressable read-write memory system. So my argument, and what I think we are now starting to see, is that this is a binary code which all animals run, and that this supports the view of the mind as an information processing system or some sort of organic supercomputer. And I've